Now, we're going to be looking at the interaction of the antibody with specific antigens. Remember here, we're not looking at antibodies and indiscriminate reactions because, well, antibodies do not react indiscriminately. They only will react to bind to proteins or antigens that have a specific shape that is able to get into the direct folding pattern of the variable region. If you remember, the variable region is up here, blue, yellow, as they're showing here, heavy chain, light chain. The white non-colored regions are going to be the constant, constant heavy, constant light. Within the variable regions, they are called variable because they are, well, because that's exactly what they are. They are variable from B cell to B cell, depending upon which antigens they are, have been elicited towards. But even within these variable regions, between B cell clones, there's going to be areas of what they would call hypervariability. These hypervariable regions they're showing here, highlighted in red. Here's all the amino acids. That's what they refer to as residues. They can be a couple of amino acids, individual or short amino acid chains within the immunoglobulin domains. The blue, the yellow you see here, yes, these are variable. The more variability, B cell to B cell, within clones, the higher the peak. But these three regions you see here in the blue, three in the yellow that are red, these are extremely variable. These hyper-variable regions take an antibody that already has some affinity towards an epitope or an antigen, and it can increase it by magnitudes. And the B cell that produces these increased affinity antibodies will get more signaling through its receptors, causing it to make more clones of itself. So this now becomes the predominant B cell making antibodies towards whatever is there, whatever is triggering this response. You see, these are referred to as HV1, HV2, HV3. They correspond to specific amino acids, 30 through 36 for HV1, HV2s 49 to 65, and 95 to 103 for HV3 on the heavy chain. Light chain, 28 to 35, 49 to 59, and 92 to 103 respectively for HV1, HV2, HV3. This increased variability, as I said, increases the different three-dimensional structures that may be produced by B cell within this variable region, at this face of the variable region. They've referred to this binding site here as the complementary determining regions, CDRs. Remember, everything here is based on three-dimensional shape. So, whatever that three-dimensional shape is, the topography of it, the ups and downs, the nooks and crannies, that's what antibodies will respond to. Turns out, these hypervariable regions are not going to be in the beta sheets. The beta sheets there are for structure. They give the overall three-dimensional shape of the immunoglobulin domain. It's in these loops, as you see here. Go from primary to the secondary to the tertiary structure. You see the loops on the face here that correspond to the binding face of the quaternary structure. These loops are the variable regions, because this is where the direct interaction between the antibody, antigen, receptor, antigen. Whatever is there on these loops is going to be exposed to the environment, and this is where things will start to interact with. When you look at these hypervariable regions, you see that the CDR, the complementary determining regions, all will cluster together. By clustering together, they will determine the binding site. They will determine the three-dimensional shape of this binding site. And if you've ever had me for another biology course, you know my favorite phrase: shape dictates function. So the shape of the CDR region is determined by the hypervariable region. The hypervariable region is determined by what is referred to as combinatorial diversity. Combinatorial diversity means that the genes can slightly mutate at the genetic level, so that you get some slight differences in the amino acid sequence in the primary, gives you a slightly different secondary, tertiary, quaternary structure within these three regions, which might give you either a better antibody or a worse antibody. The better the antibody, the better the receptor on the B cell. The tighter the interaction, the better the interaction, that B cell gets stimulated for clonal expansion. If it goes the other way, and this hypervariable regions give you a lower affinity, a lower attraction towards the antigen, well, then that receptor is not going to work so well on the B cell, which means the antibodies aren't going to work so well, and that B cell is not going to get triggered as much, and given enough time, it will just die off. So when you look at this a little closer, the regions correspond to loops. The beta sheets are structural and structural only. So we can follow this, as I said, primary to the secondary to the tertiary structure, as we see here with the beta sheets folding into the different patterns. All of the loops seem to congregate in one area, and that area will give you, as we see here, the CDR, the face region of the binding region of the variable antibody. Sorry, lost track of where my brain was going. Now, when it comes right down to it, these variable regions, like I said, are going to form pockets and grooves with the sides of these grooves, sides of these pockets, or these raised regions, going to be composed of amino acids. The amino acids each are going to have their R groups pointed in, pointed out, creating different three-dimensional structures. The epitope, or whatever this antibody will react to, bind to, can either be a linear amino acid sequence, and that linear amino acid sequence can interact with the R groups of the hypervariable region amino acids, or it can be just a chunk of a polypeptide or a part of a protein, and it's the three-dimensional nooks and crags of that epitope that just fit into the socket. If you look here at this figure 4.8 from the textbook, you get a better indication of what I'm talking about. Pockets, nooks, crannies, bigger, you get, things jetting out that will fit into the epitope. The epitope will have a specific shape that can fit into the grooves, and then the amino acids, the R groups of the amino acids in these different sized epitopes will interact with the R groups of the amino acids of the hypervariable regions. So you see here, this is a crude three-dimensional drawing by an artist. Here we see the space-filling models giving us the same thing, but here we see it X-ray crystallography from, well, actual antibodies to show you the different shapes, the nooks, the crannies, the valleys, and the projections up that will insert into the epitopes specific for, with a higher affinity for that antibody. Since we're using our groups to interact between epitope, antibody, we have to go back to many of the bonds that we've learned before. Notice here all the different non-covalent bonds we're going to be looking at. Non-covalent. So we are not getting a carbon-carbon or carbon-nitrogen interaction here. What we are getting are electrostatic forces, opposites attracting, hydrogen bonding, hydrogen to oxygen. Vander Waals, remember Vander Waals, you force two molecules close enough together, they will interact. And then hydrophobic forces, whether they're water-loving, water-hating, there's going to be water present or absent in the different pockets and the nooks and crannies, and different amino acid R chains are going to try to insert into that region.